Right now, there's a buttload of information being thrown around about the coronavirus, much of it from sources that are less than reliable. With all of the real information drowned in a sea of things Uncle Steve said after reading a Facebook post, it can be difficult and, frankly, just obnoxious to figure out what the heck is going on. Where are we really in terms of getting a vaccine for this thing? And how exactly is that even supposed to stop the coronavirus? Fortunately, Nature Journal's feature explains exactly that, as of April 28th. Right now, over 90 vaccines are in development, many of which are using brand new technologies that haven't been used in any licensed vaccine. These technologies can be divided into four different categories, according to what is injected into the body to trigger the immune response: virus, viral vector, nucleic acid, and protein subunit, each of which have two subcategories. Let's look at what distinguishes each of these types of vaccine. Virus vaccines. These are the prototypical type of vaccine you probably think of when you think of a vaccine. The virus itself is injected into your body in order to develop an immunity. Of course, the virus is weakened first. We don't want people getting sick off of what is trying to protect them. And there are two ways to do that. The first way is to give the virus a bunch of animal cells and let it reproduce on its own until it mutates. Okay, when I put it that way, it might kinda sound like the start of a sci-fi horror movie, but I promise it's not as ludicrous as it sounds. It's called a weakened virus vaccine. You see, with every mutation, the virus is essentially rolling the dice with its genome, and sometimes it loses. By isolating the virus's diroles in a lab experiment, we can choose a strain with mutation that has rendered it uninfectious, and then plug that one in the vaccine. Easy peasy. The second way to make the virus safe for injection, which is done in inactivated virus vaccines, is by simply blasting it with chemicals and heat. Turns out, if you throw enough formaldehyde and scorching temperatures at something, it tends to stop functioning properly. Imagine that. Viral vector vaccines. If you thought the last one sounded crazy, viral vector vaccines are an even more buck-wild concept. Rather than injecting you with weakened coronavirus, these vaccines consist of other viruses like measles or adenovirus that are genetically engineered to produce coronavirus proteins. It's actually quite safe and effective. Like Darth Vader betraying Palpatine and turning back to the light side, these old measles viruses metaphorically throw coronavirus into the Death Star core of the human immune system. What's actually going on is the virus from the vaccine has a gene inserted that produces the coronavirus spike protein, which is exactly what you think it is. These spiky red things. And your immune system will then respond to this protein and produce an immune system response as if it were infected by coronavirus, hence building a lasting immunity. If the virus used to produce these proteins replicate inside the cells it infects, it's called a replicating viral vector vaccine. If not, it's called a non-replicating viral vector vaccine. What can I say? Scientists are using all their brain power saving our lives from disease, not coming up with creative names. Nucleic acid vaccines. This type of vaccine decides to skip the middleman and simply inject the nucleic acid into your cells, and let them produce the coronavirus proteins themselves. As you might expect, there are two types of nucleic acid vaccines: DNA and RNA. In either case, there needs to be some way to get the nucleic acid through the phospholipid bilayer into the interior of your cells. For DNA vaccines, this is done by electroporation, which, besides having an awesome name, increase the absorption rate of DNA into your cells by blasting holes through their membranes. RNA vaccines take a page out of the Trojan horse's book and send RNA to your cells in bubbles of lipids, which merge with the cell membranes so the RNA can get inside. Once the nucleic acid gets inside the cell, it starts to produce spike proteins, which trigger an immune response, and, before you know it, your body has an immunity. Finally, there's protein-based vaccines. These vaccines trigger an immune response by containing individual bits and pieces of the virus, but not the whole thing. It's like how you can tell from just a few key lines what I'm drawing here. obviously a cute, innocent butterfly. Your body can respond to just a few key protein subunits of a virus, and hence the subcategory that uses these is called the protein subunit vaccine. The other subcategory is the virus-like particle vaccine, which, rather than including individual proteins, introduces particles that are similar to the whole virus, but hollow and harmless. When you think about it, the concept of a vaccine is a crazy one. If I didn't know any better, I'd say injecting people with the very same things that make us sick would be some kind of cruel torture, not medicine. But the amazing thing is they do work. And the fact that there are so many people working tirelessly as we speak to develop them is a testament to human ingenuity, medicine, and compassion. We can cut out a bullet lodged in someone's brain, implant a machine to keep a heart pumping, and build a robot that can replace a cripple's limbs. But when it comes to stopping a disease that attacks us on a cellular level, our best defense mechanism is ourselves.